All right. Hi, everyone. We have another session of Sybeco today. Quentin Chevy, who I know from Cold Spring Harbor when I was a postdoc, and you were also a postdoc, right? Will present today, and I let him explain more about his research and education background. Go ahead, Chevy.
Yeah, thank you, Farzaneh. It's very nice to get this email. I don't know, a couple of months ago when this paper got released on bioRxiv and got the invite to talk about it. So yeah, we met at Cold Spring Harbor. Well, I was a postdoc. I'm still a postdoc, but now the lab has moved to Washington University in St. Louis. I've been trained in Paris, where I got my PhD in neuroscience, where I was studying the mechanism for synaptic plasticity in hippocampus and the relation with inhibitory transmission. And then I moved to Cold Spring Harbor, and so now to WashU, where I'm still working on learning, but not at a system neuroscience level. And I'm still interested in interneurons and how interneurons help with learning in cortex in particular. So yeah, so this is a preprint that got released 6 months ago. So I'm going to try as much as possible to keep it in the philosophy of the journal club. So, but also because I have all the, the data and stuff, I'm not going to flash the figure. But instead, I sub-selected some panels that I think are important to the paper. So yeah, so this paper is looking at how interneurons and acetylcholine could participate in some sort of reinforcement learning in cortex. And so you have to think about this paper maybe as trying to find a general framework for how learning may happen in cortex. So I'm not really interested in the specific behavior. I'm not interested in a specific brain area, but rather it's trying to find foundations for learning, and in that case, learning driven by reinforcement feedback. So I have one slide that is very quick about a general introduction about what I mean by reinforcement learning and what's the question really and what's the gap I'm trying to address here. So reinforcement learning, being in talking to Fars in this lab, I'm sure you a lot familiar with some sort of reinforcement learning algorithm of computation that can happen. So in general, the way we think of reinforcement learning is like if you're an agent and you take an action, and in an environment, and this environment is going to give you a feedback. So this feedback is usually a reward or can be any type of a reinforcer. So it can also be a punishment, it can be, and a reward can take different form. And using this reward, the agent is going to change and adjust this action. It's going to learn to try to get as much reward, as much reinforcer as possible. So this is very, very powerful for this type of algorithm, are very powerful for decision making and action selection. Has been heavily studied in the basal ganglia, in the striatum, and using dopamine as a teaching signal. But in cortex, we actually rarely think of learning in cortex as reinforcement learning driven, and usually more as like a backpropagation type of learning, where the area above you is going to tell you whether you were right or not in processing whatever you were processing. Or Hebbian learning, which is more like something that is happening during development. So where if you fire together, you get wired together. But so why am I bringing up reinforcement learning? And maybe because that's maybe only what you need in cortex. You set up your system, your connection, your network, and then you learn through backpropagation to improve on your prediction and your sensory prediction. But the fact of the matter is that if you look at a lot of papers, you put an animal in a box and you're gonna get them to do some sort of a task where they're gonna get a reward. And often people in cortex will actually record some neurons, and we look at how the neuronal activity change through learning. And these animals, if you take the visual cortex, for example, neurons already respond to visual stimulus, but you can see that if this visual stimulus is paired with a reward, the visual cortex is going to start to encode this visual stimulus better. So this is kind of like a cartoon here that you could imagine this type of experiment where you have gradings.
Quentin, I'm sorry, we are not seeing your screen. Are you sharing?
Oh, not at all?
Yeah, so we've been
my
seeing
gosh.
you.
All right.
I think it's in a different screen, maybe.
I guess it's because it stopped when you started to record. Yes.
Right, it does that.
Okay.
Okay.
Do you see it now?
Yeah, and can we see your first slide?
You're good here?
Yeah, we haven't seen anything, so if you want
Okay,
to...
that's all right. That's all right.
Yeah.
Can
we see the first slide, please? I just want to make sure we have the very first, the cover slide.
Sorry about that.
Okay, thank you.
So that's just the very first slide. Again, I'm trying to we can just look at the bottom here. So yeah, as I said, if you are studying visual cortex and your favorite, part of cortex is visual cortex, you could imagine an experiment, and this experiment has been done many, many times, where you have an animal and you pair some sort of gradings moving one way or another, and you look at the sensory tuning properties of neurons in visual cortex. And you're going to see that these tuning properties are going to change based on whether the visual stimulus leads to a reward or not. So the question is, what's the teaching signal that drives this type of learning? Okay, how like this cannot be explained by just simple bypropagation or Hawaiian learning. What's the feedback that gets to the visual cortex that will influence sensory tuning? And you can actually find this type of example throughout the cortex, whether it's auditory cortex, somatosensory cortex, and so on. I think the biggest problem, and why maybe people never ask this type of question in the context of reinforcement learning, is that when you're going to record a neuron in the visual cortex and you're going to have a visual stimulus, you're going to have a confusion between the cue that you have to learn that predicts a reward and the latent feature, which is like this cue actually predicts a reward. So you have a sensory stimulus, which is what's being encoded locally, and you're going to have some latent features on top of that sensory stimulus. So some people, would study attention, some people will study movement. In our case, we're going to study reward and reward predicting cues. So that's the kind of framework I'm trying to bring here is, and the question, this paper is asking is, can we identify neural correlates of reinforcement learning in cortex? So can we find a reinforcement feedback that arrives to cortex and that could change neural activity? And so this paper focuses mostly on this microcircuit that, again, as being in Farzane's lab, you must be very familiar with, where one of the big hypotheses that the reinforcement feedback is going to be brought up by acetylcholine inputs, specifically targeting VIP neurons, and this disinhibitory loop through VIP interneurons and somatostatin can gate plasticity of principal cells and promote learning. So yeah, so I'm going a little quick here because again, it's I'm taking it as a journal club, so I want just to go into the data and like the figures and talk about the figures with you. But that's kind of like the main claim that we're gonna work through, are the fact that the local and global activity of VIP neurons. So what I mean by local is like if you're in the auditory cortex, VIP neurons will respond to sound, and the global activity will be a reward or a cue predicting a reward is what I call global. So the local and global activity of VIP neurons is modulated by expectation, by the fact that you can expect the delivery of a reward or not. That acetylcholine provide this global sensory modality independent signal to cortex, and that somatostatin and recurrent connection between somatostatin and VIP allow to gate the direction of the flow. Do you learn from your feedback, or have you learned already and you can stop learning? And then we have a model to like kind of formalize that statement. One last thing, and then please like interrupt me whenever he wants, and we can look through specifics in the figures. But as I said, the problem when you study cortex, which you don't have when you study dopamine or subcortical area, is that cortex does something locally. Okay, if you are in auditory cortex, you are going to code for cues, for auditory cues. If you're in visual cortex, you're going to encode for visual cues. And so how do you separate your sensory coding from your global cue, typical cue coding? So this is what the paper, in terms of its methodology, is organized around. Is trying to manipulate sensor modality to separate the local sensory coding from the global general reinforcement coding. So this paper used principally two different tasks. One, which is what I call an auditory task, where there is a sound that 80% of the time predicts the delivery of reward. Okay, there's no, it's purely Pavlovian. The animal doesn't have to do anything. It can just listen, and then it's going to learn to predict the reward. So you're going to see the animal starting to lick in anticipation for the reward. And we're going to compare this sensory stimulus to a neutral stimulus, which is never followed by a reward. And we're going to compare it also what's happening to the reward response when the animal can predict the reward versus cannot predict the reward when the reward is uncued. And the second task is exactly the same, except that now instead of a sound, it's a left light stimulus or right light stimulus that predicts a reward with the same probabilities. And so in that case, by manipulating the sensory modalities and recording, in our case, in auditory cortex, you can actually separate the local coding from the global coding. And as a segue, this is kind of also what this paper is organized around. There's two types of recording. One, which is vibrophotometry in auditory cortex, where we're going to record population of BIP neurons or somatostatin neurons or acetylcholine release in auditory cortex during these two cues. And so that will be where you can access the local computation and how it interacts with the global computation. And the other setup is using two-photon microscopy in parietal cortex as a non-primary sensory areas. Although I'm sure you're all aware that actually the parietal cortex also encodes some sensory features. But yeah, so that's kind of like the methods here to separate local sensory activity from global reinforcement learning type of feedback. One last thing is why BIP and why I'm interested in these particular neurons is actually this work is a kind of on the shoulder of two main paper. One which has been done by the two that have been done by our lab. One that shows that BIP neurons in auditory cortex indeed responds to reward and punishment, where we use electrophysiological recording to show that, where you see that if you give a reward or punish the animal, you see this increase in spiking activity of BIP neurons. And the other one, which was kind of a follow-up of the study that I participated in, where you show that if you record BIP neurons anywhere in cortex, you're going to see that they respond to reward. And so that led to the question of, like, if they respond to reward, can they actually be involved in reinforcement learning and start to respond to cue predicting rewards and what's happening to the reward response? All right. Let's go directly to the figure. So figure one, sorry. Figure one is really trying to jump directly into this problem and ask, what's happening to this reward response from BIP neurons when the animal can predict the reward? And so figure one is using this task that I described, where there's a sound that predicts the reward, that we're going to compare to a sound that doesn't predict the reward. And we're also going to compare what's happening when the animal cannot predict the reward. This is the leak rate. This is the only time we're going to talk about leak rate, just to show you that the behavior, we primarily also a behavior lab, so we pay attention to the fact that behavior should be good. So here in case of behavior, we're talking about that the animal is developing this anticipatory leaking. So it's going to leak before the reward happens, more for the cue that predicts the reward than for the cue that doesn't predict the reward. And this is the type of recording we're going to look at a lot, which is in that case photophotometry. So we're using VIP-3 animals and injecting GCamp in auditory cortex, inserting a fiber to record GCamp related activity from VIP neurons. And you can see that changes in fluorescence kind of give you an idea of what are the population coding from VIP neurons.
Question, Quentin. So this is a typical condition, classical condition in tasks, meaning
Yes.
that the stimulus comes, then the reward. So the reward first comes and then the mouse licks, right? It's not operant.
No, it's
Condition.
not operant. There's nothing to do. Okay, it's
Okay.
So what we it's a trace conditioning. So there's a cue, a 1 second delay, a reward. Okay, there's no and so
And how long is your cue
it's a half a second.
for both auditory and visual?
Yes, yes. We
Okay.
try
And
we try to keep everything as much as like the same.
Right, and there's always 1 second gap. All right,
Correct.
thanks.
Except for one experiment, but with the 2-photon microscope, but that was more for technical reasons.
Right, and then is it like during this 1 second, they don't like until the end of the 1 second?
So animals doing this Pavlovian conditioning, it's I think the animals have like animals have different strategies. Some of them leak only during the cue and then stop leaking. It's rare. Stop leaking the delay period. Others kind of like ramp up the leaking to the reward. So it's kind of yeah. Every animal is kind of the same. But here we're measuring the leak rate in that case, like during the delay period, because we found that this is where you can consistently have this like difference between conditional stimulus and neutral stimulus. All right. So I'm gonna try to keep, because I think one of the feedback from the reviewers was that it's hard to follow the change in conditions, the change in task, and the change in where we're recording. So I'm gonna try to follow their advice. And here on the left, often you're gonna have a reminder of what's the behavior and where we're recording and what we're recording. So here I'm going to show you some example trials and then an average plot of VIP activity early during learning of this task and then late after learning of this task. So early is actually the very first day. So the very first day you put the animal in the box, and then you record them during just the exact same task that they have to learn. So there's no change in trial types probability or anything. It's the first time they hear those sounds and paired with reward. So what you can see is that here the color coding is that blue means no VIP activity and yellow means a lot of VIP activity. And so what this plot is showing is that in auditory cortex, VIP respond to sound when the sound is being played, and also respond to reward here, like uncued reward. Okay, so you have a sound, a reward, and so that's the cued reward condition. And here you have an uncued reward condition where there's no sound being played and just the reward. So here it is, in essence, nothing new. VIP neurons in auditory cortex respond to sound, and VIP neurons in auditory cortex respond to reward, which is something we've been documenting for quite a while now. Okay, what was very interesting to us and kind of led to this whole paper is that after learning, something very striking happened. So if you focus on the middle plot here, you see that the reward response, the uncued reward response, is still there. So after learning, when the animal cannot predict the reward, the VIP neuron still responds to reward. But after learning, when the animal can predict the reward, they completely stop responding to reward, or they have a decrease in reward response, and they also have a decrease in auditory response. So it seems that the conclusion of figure 1 is that both the reward and the sensory response from VIP neurons goes down with learning. okay, which is something that was unexpected. But also, you know, if you're an aficionado of stuff like intent neurons and learning, you actually often you see that VIP neurons don't respond very well to sound and local inputs. So, you know, but that's something we can discuss.
This is beautiful, by the way, clean results. So this is aligned with surprise coding for VIP neurons.
Yes.
And did you guys by any chance see any prediction signals like during the trace time, during that 1 second?
What do you mean by that?
Some anticipation of the upcoming reward.
Well, so here the animal anticipate the upcoming reward.
Correct. And I'm saying, did you see any VIP representation of that?
No. No, not in this simple task. Maybe we have something in tasks that are a little bit more complicated, and not in this setup. The figure 2 is actually the one that shows that actually they do. So yeah, because I think what's important here and what has been missing again is this like conflation of global and local activity, and it's not dopamine. Okay, you know, for those here who know about dopamine and reward prediction dopamine, it's harder, like we cannot translate that completely.
And how many VIP neurons are we looking at here with fiber? Is it like hundreds?
Unlikely it's hundreds. Hard to know.
Tens?
Yeah, honestly hard to know. But we have a single VIP neurons then later on. So when we look at somatostatin neurons, which have like a much better signal to noise ratio, even with fiber photometry, I think you could imagine kind of measuring how many neurons you're recording under your fiber. I think it's a tricky question, fiber photometry in general, how many neurons you record. One last thing from this figure, and I put that here, it's not very important, but actually it's an experiment that I like a lot, is so this animal I've been trained on this task here, but the first day of this task, they learn how to lick to get their reward, and I'm also doing an auditory tuning. So I'm playing different sounds. So the chirps, which are this sweeping sound that goes from 4 kilohertz to 20 kilohertz, or the other way around. And that's what the animal have been trained to associate with a reward or not. And I'm also playing pure tones that this animal will never hear these pure tones ever during the task until the very end when they learn the task. When I decided they've learned the task, they have this unspectral leaking, and then I'm running this auditory tuning again. So if you read, and I'm sorry, there's actually a typo in the paper about the legend here, but this is the correct legend. What you see is that actually the cue response, the sensory response from VIP neurons that have been associated with reward goes down. And both of them actually, both the conditional stimulus and the neutral stimulus, and we can discuss why it could make sense in terms of generalization, in terms of like what's the demand of the task. But sound that have not been played during the task actually are not impacted. They're not affected. Okay, so it seems that the loss of sensory response by VIP neurons is specific to the sound that have been used during the task and not other sounds. And that's pretty important. And that's kind of going to help us trying to like model and interpret the data. Yeah. Everyone's following?
Yes.
Okay, so as I said, one of the tricks that I like in this paper is the fact that we're trying to decouple the local and the global signal, the sensory information from the cue information. And one way to do that, as I said, is to remove the sensory information, the sensory drive from your recording. And so what you can do is you stay in auditory cortex, and instead of pairing a sound with a reward, you pair a visual stimulus with a reward. And this is what exactly figure 2 is about doing this type of experiment. And we can discuss also surprise coding in here. But what you see again is the exact same type of plot that I showed you for figure 1. It's early on in training. So the first day, this animal, see these slides turning on and get a reward. And you can see that, so here in this example, we don't have much of a visual response, but we have this reward response because the animal cannot predict the reward. So it's like an unqueued reward, VIP response to unqueued reward, or unexpected reward in that case. But actually, if you look in average, you see a small visual response, which we think is some sort of saliency or some sort of surprise response from VIP neurons in auditory cortex. And it's being discussed in the paper. After training, what you see is a little bit what you were talking about, Porzene, is you see that the reward response goes away. So now the animal can use the cue to predict the reward. And now the reward is not surprising or can be predicted, and therefore is not being encoded by VIP anymore. And instead, now VIP start to encode more strongly the cue that predicts the reward. Now you're more into a typical reward prediction error coding, where when you can predict the reward, the value of the reward goes down, and you don't need to encode it anymore. Instead, you code for the cue that predicts the reward, and the neutral stimulus, one that would not predict the reward, is not being encoded. And so by this sensory modality trick, we can reveal a reward predicting coding by VIP neurons in auditory cortex. You can do the other way around. Instead of changing the sensor modality, you can change the brain area, but keep the auditory stimulus. And so that's what we did with some collaborators, and also the co-first author of this paper, Zoltan. Where here we also, that gives us an idea of the
question, Quentin.
Yeah.
Okay, so could you go back?
Mm-hmm.
So how come is it that they respond to the predicting stimulus if it is visual, but not if it is auditory?
So that would be kind of like figure 6 or 7. Like,
Okay.
what I think that's the the next stage is like, why? So who is informing VIP of the presence of a reward or a cue predicting a reward, and who is inhibiting VIP locally?
Yeah,
So,
and the other interesting thing is that the type of response to the visual stimulus here after learning is very much of an evoked response. But the type I had in mind is more of a ramping anticipatory response, like a CR, like reflecting that.
I think
Like,
it's a tricky subject, even in the dopamine field. If you look at the dopamine, most 99% of the report, you would not see a ramp. To see a ramp to the reward, you actually need to be in very specific conditions.
right.
Most often, you see a cue response, and you may see that it goes, it continues during the delay period. But actually, to see this ramping, you need to go single units and do some, maybe complicated thing to reveal the ramping activity. So I'm not totally surprised to not see this ramp. But yeah, I think maybe you're referring to something else, but.
Yeah, for example, in the cerebellum, we see it more like a ramp in Purkinje cells, but
Yeah.
also whether predictions are represented as a ramp or as some other type of response. I mean, ramp is just one way of neural representation of predictions. It could be in this way. It could be just like a salient invoked type response. Yeah. Okay.
Okay, so yeah, as I said, now we're doing kind of the opposite experiment where we just changing brain area but keeping the sensory the same sensor modality. So here a sound, and we're also using 2-photon microscopy thanks to Zoltan, and that gives us access to also kind of the heterogeneity of VIP neurons. But we already had a paper about the heterogeneity of VIP neurons, and kind of the conclusion was like, 80% of VIP neurons respond to reward. So that also what allows to do fiber photometry in a way. So here it's a small panel of this figure to show the single cell reward response from VIP neurons, and using a simple threshold based at like 3Z score, which is kind of classic threshold to decide whether something responds to not. You could get the same result using a K-mean algorithm of some sort. But you can find that, again, we found that the vast majority of VIP neurons in parietal cortex respond to reward, and there's this like subgroup that doesn't. And if you now look at so now there's no more like learning early and late, because it's a 2-photon experiment, so it's a little harder to do that chronically. So here I can only show you after the animal has learned the task. What you see is that the VIP neuron responds to the auditory cue that predicts the reward. And actually, the subgroup of neuron that doesn't respond to reward doesn't show a cue predicting response as well. And these neurons also barely respond to the neutral stimulus. So here it's leading to the same conclusion that once you remove the sensory drive, you can reveal a reward prediction error type of coding by the IP neurons in cortex.
I got a bit confused here. So is this figure after learning?
Yes, it is after learning.
Okay, and then and did we see the figure? Okay, you said you don't have the figure before learning.
No, we don't have with 2-photon. We don't have before learning. We train the animal and then record them once they've learned. So the only thing we can compare is across the different trial types. So we can compare a neutral stimulus against a conditional stimulus.
And this response seems to be very sustained, up to 4 seconds after the reward.
Yes, it's something we see in this batch of animals, that the reward response is very long. Don't really know how to explain that. Although the acetylcholine data kind of goes towards the direction of something that is more like long, but there's a lot then there's a lot you can discuss about the transduction from an acetylcholine input to a VIP neuron input, the type of receptors are going to be used and stuff like that. So I think it's something that could be expected, but
okay.
All right, so I think that's a segue a bit for the next figure. So I hope these 3 figures kind of demonstrate that there is a reward prediction error coding by VIP neurons in cortex, but as soon as you add some local sensory activity, things are getting a little more complicated. So the 2 questions the rest of the paper is trying to address is what drives VIP neurons to encode reward prediction error, and what locally makes this coding a little bit more complicated once you have this sensory drive. So figure 4 is a little bit trying to motivate why we're going to look at acetylcholine. The first motivation is by using the same technology, 2-photon microscopy, is to look at acetylcholine release around VIP neurons. So here we're using an acetylcholine sensor, which is called GrabACH. So this is written here. So it's a recombinant receptor that will fluoresce if you have acetylcholine being released in the environment. Okay, and the way to probe the connection between acetylcholine release and VIP neurons is we also, in the same animal, have label VIP neurons using TD tomato, and so we can look around VIP neurons, their fluorescence from the GrabACH, and see whether acetylcholine is being released with at the vicinity of VIP neurons. Unfortunately, we cannot make in this figure here, a stronger statement about that, because we don't have a cre-dependent virus at the time. So that, anyway, I don't know if any one of you or has ever looked at acetylcholine release in Cortex, but this is very, like, this is flooding the zone. It's
Very
like
interesting.
everywhere.
Quentin, question. So this biosensor, so you have that and then you inject GCAM, so you have both the output and the input kind of.
Here, no. Here we only have GrabACH being expressed by cortical neurons.
Okay. So GrabACH is an acetylcholine sensor.
Yes, correct.
And it's expressed, how do you limit it to cortical? So you just inject it there?
Yeah, you just inject and record where it's being injected.
And then VIP to the tomato, the idea is to, how do you use the VIP label in here?
So you just look at the green fluorescence from the GrabACH around this little cube that we made as like an ROI around VIP neurons.
I see.
We have, like in the supplementary figure, there's an attempt to do, I mean, I'm saying attempt because I think the virus is the expression is very low, but
But how sure are you that this cholinergic input is onto the VIP neuron?
Yeah. So figure, I don't remember the name of the figure. I'm sorry. One of the supplementary figure, we actually express GrabACH in a tree-dependent manner in VIPs. So that's panel E, and we see that when you deliver a reward, you would see an increase in acetylcholine on VIP neurons. But this is very small, and I think it's just the virus doesn't express very well, and so it's, you know, you were asking how many neurons you record even less when you do that. But we try to have different ways to show this connection. One is by manipulating acetylcholine inputs to VIP and show that, you know, if you inhibit them, you see less of a reward response. Now I'm getting another figure ready to show that if you activate acetylcholine input onto VIP, you get a VIP response. So we're trying to cover the link between acetylcholine and VIP in different ways, but also it's something that's been shown like many times, and so we don't have to.
And then why did you guys went with GRAB instead of measuring cholinergic inputs in the cortex?
With what, GCAM?
Like chat-cree, yeah, GCAM.
Yeah, GrabACH works extremely well. I think it's just like the signal to noise ratio is amazing, and we started to use that for, you know, many different projects. So that was kind of like an easy
and
way.
Just to, I'm sorry you said this, but this is a virus injection?
Yes.
Okay, cool.
So the take-home message of this figure is that, you know, it's a correlated figure, but is to say that if you record GrabACH next to VIP neurons, you see that they're going to be an increase in acetylcholine release upon reward. So that's the panel B here. Wherever you look, you know, on whatever VIP neurons you're going to look at, you're going to see this increase in acetylcholine release as seen as an increase in fluorescence at the vicinity of VIP neurons. And at the time, we didn't have two-color fiber photometry, so, you know, I tried to like hack my way around that by doing bilateral recording of on one side VIP neuron and on the other side acetylcholine release, and showed that these signals are very correlated. So when you have an increase in VIP neuron activity upon reward, you see the same increase or with the same latency in terms of like release of acetylcholine. And so, yeah.
Is there spectral overlap? Is that why you don't do it in the same?
At the time, it's just because we didn't have the, you know, the red and green. But no, there's no. I mean, the spatial overlap can be remedied, so it's not, you know. Now we're gonna move towards two-color fiber photometry, but at the time we just couldn't.
So GRAB is what color is GRAB?
GRAB is green. There is a red GrabACH that just got released 3 months ago.
And it's excited by the same wavelength as GCAM, or do you need a different excitation?
No, you would need. I mean, you could You could excite them using the same wavelengths, but then you run into issues of crosstalk. So you wanna use different wavelengths. Yeah. So again, I'm gonna go a little fast on figure 4 because I think it's just and the supplementary figure that goes with it, because I think it's just a way to motivate the link between VIP and acetylcholine, and that's a link that's been done many times. So we didn't want also to redo everything. Yeah, so figure 5 is here to demonstrate that acetylcholine is indeed capable of providing a reward prediction error coding to cortex in a sensory modality independent way. So here we're gonna focus on recording acetylcholine release in auditory cortex using fiber photometry in this auditory and visual task. And basically, the idea is like, we could discuss details and like what's the difference between the two tasks, but the idea is like we see the same type of reinforcement learning related activity by acetylcholine release in cortex in the two tasks. So what I mean by that is like early on you see that an unpredictable or unpredicted reward leads to a release of acetylcholine in both tasks, and then later you see that this release of acetylcholine moves to the cue. So either auditory cue or visual cue, and again we are in auditory cortex here. So you see a release of acetylcholine in auditory cortex, whether you have an auditory cue or a visual cue. And there's a supplementary figure that showed that it's even the case if you use an olfactory cue. You would see an acetylcholine release to cue predicting rewards.
So am I saying this right, that the pattern of acetylcholine activity is similar to VIP for the visual stimulus, but not for the auditory stimulus?
Correct. Yes. Yeah.
And you're coming back to this?
Yes.
Okay.
So here the take-home message is that acetylcholine provide a reward prediction error coding to cortex in a sensory modality independent way. And because acetylcholine activate VIP neurons, or the activity of acetylcholine release correlates with the one of VIP neurons, we can assume or conclude that the reward prediction coding from VIP neurons is being inherited from acetylcholine. But now again, what Farzaneh, you brought up is like, what's happening during the auditory case? Why do we have this inhibition? Why is it that VIP neurons don't follow acetylcholine? And so that's kind of like what I've been hunting for the past couple of years. And now my focus is like switching a bit on somatostatin interneurons, and that's what you may realize in the paper.
Is it not surprising that acetylcholine is about arousal? So I would have guessed that any salient stimulus would increase the acetylcholine. So how come early on we don't see any responses to it?
So you do see actually a response, and I think that's the important part. I mean, there's different ways to think about it, but first, like if you look early on, you have a little bump. You have at the moment of a sensory surprise will lead to a release of acetylcholine.
But not in the auditory case.
In average, you see something happening. If you look, obviously, these are like example animals where I'm trying to like, bring a message home, but in average, you would actually see some saliency response. But what's important is that the saliency response goes up with learning. And this increase is specific to the cue that predicts the reward, not to the cue that doesn't predict the reward. So that's one argument for like, this is not just, a surprise response, because actually this response goes up if you predict the reward. But then you can always make an argument that is about arousal, and arousal is going to go up with the reward. And it's hard to separate you arouse because you have been reinforced. So it's as soon as you touch the cortex, people want to talk about arousal. And we have some like running behavior and like pupil to like show that whether the animal is running or not, we're gonna see the same response. Whether the animal has a big pupil or not, we're gonna see the same response. We're gonna see the same type of reinforcement coding. That being said, I think obviously acetylcholine is coding for arousal. So I'm not saying this is not an arousal type of response, but maybe we could also reframe that in terms of reinforcement learning.
Okay.
Yeah, so in a way, this paper is trying to end by looking for what inhibits VIP neurons locally when there is a sensory drive. And so this connection between VIP and somatostatin leading to disinhibition of principal cells and getting learning. At the same time, people show that somatostatin also have feedback inhibition onto VIP. And so this somatostatin is actually kind of the major source of inhibition for VIP. So that's where I started to look at like what's happening during somatostatin during that task, and can this explain what's happening to VIP neuron. So the first thing I did was to record somatostatin in the auditory cortex using the same approach, using fiber photometry. We in auditory cortex, so if you play sound, you see this beautiful sound response from VIP neurons. But then something is striking, and I was not expecting that at all, is that I started to find that some animals, so which we can discuss what does it mean when you do fiber photometry, but some animals show a strong response to reward. So somatostatin in auditory cortex respond to reward in a delayed fashion, and there is a supplementary figure 8 or 9, which is about quantifying the fact that this is delayed and stuff like that. And I think this is very interesting and fascinating, but I think it's not about, this paper is not about that. But some animals show this like nice response to reward, and others didn't show any response to reward. This is completely independent of whether they respond to auditory respond or not, to sound or not. So there's like in this figure, there's something showing that the it's not that the data is bad in this animal. That's why we don't see a reward response. And what we think actually is that just with our fiber photometry, just by chance, we are subsampling different population of somatostatin that I labeled in the paper. Oh, I realized there is a mistake here. That I labeled in the paper P and N. So P for positively responding to reward, and N for negatively responding to reward. And we kind of like, again, went back to the 2-photon microscopy in parietal cortex and look at, so actually in the paper, it's in the visual cortex, but we're moving that back to the parietal cortex. So here it's in, I'm going to show you data from the visual cortex. Yeah, so if you look at single somatostatin neuron in visual cortex, you're going to see the same thing. You're going to see that some neurons respond to reward very nicely, and others don't, and actually see some sort of inhibition. And it seems that they're located in different places in cortex, and so could represent this like microcircuit of VIP inhibiting somatostatin, and then there's a feedback through another type of somatostatin onto VIP.
What is F and S?
Sorry?
You have F, SST, SSS?
Yeah, it's because this came in work better when we asked for 3 clusters, and you could actually see fast responding and slow responding somatostatin, but most of them show this slow response.
Okay.
So yeah, so that's the final experiment in the paper. So we're gonna do the same thing as we did with VIP and acetylcholine, but this time with somatostatin. And here I separated my animals in 2 batches. one that respond to reward and one that don't respond to reward. And so here, if you look on the top, you see that this somatostatin respond to the sensory cue, so to the sound in auditory cortex, and they also show this reward response. They actually never respond to the visual stimulus, but they still respond to the reward. I mean, this animal they've been selected for the fact that they respond to reward. But what I think is fascinating to me is that despite the fact that they respond to this global signal, which is a reward response, they don't respond at all to, just a salient random stuff, which is here, this visual stimulus. And after learning, what I found is that this somatostatin increased the response to the sound that predicts the reward. But they also ramp this time, far as I know, after the visual stimulus that predicts the reward. So there's still no cue coding. So these neurons still don't respond to the cue, but instead they ramp the activity to the delivery of a reward. And I think this could have a lot of implication in terms of computation and model, and how do you actually implement that in a model that we didn't do at all. But I think this is where to go. If you take the other population, the one that don't respond to reward, you see the opposite. You see that this neuron decrease the response to cue that predicts a reward, whether it's locally encoded sound or externally, encoded somewhere else visual stimulus. And you actually see this sustained inhibition during the whole period of a visual stimulus that predicts a reward. And so what we think is that we try to create a toy model where we have, we're trying to make a biologically plausible model and not just a soup of interneurons and principal cells, to see how this microcircuit, this BIP, this disinhibition and then feedback inhibition could gate learning, but also close learning once things have been learned. And so the idea is that you have this global signal that just tells the cortex, here, something important happened. Here, a cue predicting a reward happened. And that everyone should be aware of that. And BIP and somatostatin are the one locally that would gate learning. And once things have been learned, BIP is shutting the door to this global signal. Say, okay, now we've learned, you don't need to learn anymore. So we have this model that it's pretty simple, where we are inputting an acetylcholine signal that's going to increase over time. So you have to imagine we are the moment of the cue. At the beginning, there is no acetylcholine, and then the acetylcholine is going to start to be released during the cue period, and it's going to stay. That's the important part. The global signal never change. The cue, you predict a reward, you want to know. VIP are going to increase their activity to gate learning, to trigger learning, but then VIP are going to have their activity going down once locally you've learned. And somatostatin neuron, the one that being targeted by VIP, actually going to stay down and maybe try to go back. And the somatostatin that provide this feedback inhibition kind of provide the sensory drive to VIP neurons. So are telling VIP neurons what is it that the cortex locally knows or not. And so somatostatin with the cortex learning to predict the cue, this subgroup of somatostatin are going to inhibit VIP neurons. And using this very simple model, we can recapitulate both the behavior of all these protagonists during an auditory task where there's again this sensory and global signal that interact with each other, or a visual task where they're decoupled. There's no local drive, and so VIP neuron just provide this typical reward prediction or encoding. And then we did some ablation experiment where we remove one of the actor and show how if you didn't have this feedback inhibition loop, you have some runaway happening where you just learn too much and then you just ended up losing your information, or you will learn much slower if you don't have this inhibition happening. Yeah, so that's kind of the take-home message is we focus on this on this microcircuit here to provide some sort of an implementation of reinforcement learning in Cortex. And the idea is that when you learn, you have this disinhibition happening where acetylcholine will drive the IP that will inhibit somatostatin and drive learning of local information. But once you know a feedback loop, inhibitory loop is going to come and inhibit VIP until, and stop the acetylcholine signal to be propagated to the principal cells because you don't want to run away, you don't want to become epileptic or whatnot. Yeah, so sorry, during my talk, I have this little quotation from Kundera about how you learn, but we're gonna pass that. And yeah, I'd like to thank everyone. It was a very much collaborative work between the Balazs lab and Costa's lab and Loreen in Berlin for the model. So yeah, thanks everyone.
Awesome. Thank you, Quentin.
Mm-hmm.
Could you stay here? So the middle is before learning happens?
Yeah, no, it was during learning.
And that's when acetylcholine and VIP are active, but not. But is that what you showed with your data?
What is it?
So early on, I guess I'm confused because is this model true for both auditory and visual tasks?
Well, you have to imagine, I mean, this model could be true for the visual task if you assume that for the visual task, you would always be stuck here in the middle for the visual task because you're trying to learn something, but locally there's nothing to learn. So this is saying this is gating learning. So this is allowing learning to happen, but you have no features to learn locally. So you're stuck here. Your auditory cortex will never learn, or you never know something, or you will never know how to compute visual stimulus, except, strokes and stuff. But
and then you're saying that's why, because the key difference was that with the visual, initially we didn't have the activity, but later on we had it.
mm-hmm.
And how does this model explain why initially we didn't have it, but later on we had it?
Well, this model explain how VIP tracks acetylcholine. So it doesn't explain how acetylcholine create is reward prediction error signal. But I think, it gets, you could make the same, you could ask the same question for dopamine, like how is it that dopamine creates a reward prediction error signal? So this model explain why sometimes you track VIP neurons, sometimes VIP neurons activity tracks acetylcholine release, and sometimes not.
Right. And then what is this learning and knowing? I'm not following those arrows.
It's the direction of the flow. So you have a flow that goes from acetylcholine VIP somatostatin. So this is disinhibition and learning. And here, once your principal cells have learned and the plasticity has happened, so they've learned their local features to improve their detection. Now you know. And so now you have this feedback inhibition that inhibit VIP neurons.
I see. Okay. And that's after learning. That
That's after
VIP
learning.
is.
And if you see, I mean, and I'm sure I'm sure you like have some data on that either from the Allen or I mean, there's plenty of other people who've done that, but like a lot of two-photon data are in motivated behavior are after the animal has learned, and you see that, for example, VIP neuron in visual cortex, they don't respond to visual stimulus much after learning, and we think it's because it's not that the computation or the role of VIP neurons for sensory computation is, you know, is about them being inhibited to improve some sort of like feature detection of things, but it's rather like they're there to like gate learning, to say learn or don't learn, and not to like, I mean, obviously somatostatin and VIP will participate to some lateral inhibitions and contrast enhancements, but here the idea is like to focus on learning mechanism and not necessarily on.
Yeah, okay. So I love it. You're bringing this up. I was at the Allen Institute. I'm a co-first author on the paper you're referring to. So, and it is true. Early on, we had a lot of visual responses, and later on, we don't have it in the visual, in the VIP. In my lab, we're seeing not exactly like that, but we are doing now PPC. Allen was all visual cortex. But
Yeah, so in the
model,
PPC, that's the opposite. That's exactly what, you know.
so what do you see in PPC?
I mean, like non-primarily sensory area, my
Yeah.
prediction would be that VIP response would go up with learning.
At least we're seeing that it's not going down. So we are
That's not
not
going.
seeing what we had at the Allen.
Yeah.
And then, but how do you reconcile that with your model?
Is that locally, because you're in visual cortex and you've learned your visual scenes, so you've learned all of that, so you don't need to have VIP neurons anymore to say, hey, learn. Okay.
Yeah.
So you have this feedback inhibition from somatostatin onto VIP. So you've increased the activity of this population that's going to inhibit VIP and you know, yeah, gate or turn off learning in a way.
Yeah. But then, and then we can think of this perhaps like the pyramidal cell learn the prediction, and then through the SST, it conveys that to VIP. So VIP doesn't respond to this sensory input that comes through the acetylcholine.
Yeah.
But for the PPC, then why would you say that it makes sense to you that the response will go up with learning?
Well, you know, eventually, you know, then you can ask a question about what's the local computation that the PPC is doing. And eventually, if the acetylcholine computation matches the local computation of the PPC, you may see the two canceling out each other. But as you know, a first assumption, you can say, okay, it's a locally, what is the PPC coding? You know, I guess that's what you're trying to figure out. But because the acetylcholine, the arousal or reinforcement type of coding coming onto the PPC is not going to match what locally the PPC is doing, then it means that VIP neurons are going to continue tracking acetylcholine. The activity of VIP neurons is going to track the reinforcement prediction error coding of acetylcholine.
So if I understood you correctly, you're saying that in a multisensory higher order area like the parietal, unlike the primary visual VIP, the prediction that the pyramidal cell provides is not for PPC. The prediction that the pyramidal cell provides is not an accurate prediction of what the acetylcholine representation is about. So as a result, we don't quite have this inhibition.
Yeah.
And so
yes. That's what I'm going to. I think there's a lot of limits to these statements. So, you know, I don't want to make that an absolute statement, but that would be a working model.
But the reason you're saying that is because it's not specialized for sensory inputs, right?
Yeah.
So then well, that's an interesting way to put it. It can also easily go in the other direction, that it's more of a higher order area that has access to higher level predictions. But I agree, maybe like the prediction here is a very simple prediction. It's about the visual stimulus. But if it was a broader prediction, then we would see the opposite, that it doesn't go away. Very interesting. And does this, so that's how you see it, that why for the auditory stimulus, then, well, for the auditory, but then would that also apply to here? Because I got confused with the auditory visual and the two areas, right? And you're showing one single model here. So I also, if I remember correctly, you showed us that in the auditory cortex, when we record from the auditory cortex, visual stimulus early on does not evoke a response, but later it does.
Mm-hmm. Mm-hmm.
And then I guess my question is that how do you fit that to this model of the acetylcholines?
Well,
So
acetylcholine is agnostic. Okay, it doesn't know the acetylcholine, and the same way dopamine. Dopamine just flood the striatum with a reward prediction error coding. It doesn't care about where it's going. It's just like, here, MSNs, deal with it. The same way here, acetylcholine say, here, cortex, deal with that. Do something with that. And VIP are there to first you don't want maybe to have acetylcholine dumping, this information directly into principal cells. So you want maybe some interneurons layers in that. And VIP, they disinhibits principal cells, so they would get learning. But so if learning doesn't happen, then VIP will never get inhibited, will never get this feedback inhibition. And learning doesn't happen in the visual stimulus case because the principal cells, they don't care, they don't
but
learn.
what is surprising is that they didn't respond early on. I guess that's there was a little bit of confusion here because the example heat map cell that you showed, heat map session, I suppose, showed no response early on, but then your average mice, I guess that was the average mice line plot that did show. So maybe I should not go with the heat map example as much because that's in my mind that showed there was no response to the visual stimulus early on.
No, I think obviously, that would be a fair, you see a small
but this
response.
doesn't match the heat map, and that's a little bit of confusion because the heat map shows there is no response.
You see a small response, but we think, and it's why it's not in the heat map. It's because we think this response is in the saliency realm of things. Okay, this response early on is just a saliency thing. That acetylcholine does saliency.
Correct.
Okay? Like acetylcholine is going to respond to everything and anything except things that you can predict.
Right. And then for the visual cortex recordings, you again had both stimulus types, correct?
We don't have visual cortex recordings.
But I thought you had the 2 photon.
It's in the parietal area.
Oh, that's what I mean. I mean parietal. So in the PPC, you again had the 2 stimulus.
No, we only have auditory stimulus because
I see.
It's harder to turn on the light in the 2 photon rig in our case, but yeah.
Right. Yeah, we are doing a lot of seedling. It is doable, but yeah, correct. So, but did the auditory response go away after learning in the PPC?
No,
it didn't.
It goes up. I mean, we don't know after learning because again, we don't have before after. But what we see is that VIP neurons encode a sound that predicts a reward, but don't encode a sound that doesn't predict a reward. So
yeah, that was the plot that you showed, that the auditory response. Well, you didn't show pre-learning, but after learning anyway, they respond. Okay, very cool model. And then
I think related to your work, it's kind of, I mean, maybe your past work. Now I don't know what you're going to be doing with the cerebellum, but
we actually
I think he moves the
looking at
goalpost
and parietal, so we have a lot of VIP work also going on.
Because a lot of the literature is about sensory prediction error. It's about, and which makes sense. Like the cortex is about this predictive box, and you want to predict your sensory flow. But here we're talking about reward prediction error, and how is it that you prediction, your sensory information is going to be influenced by the fact that you're going to receive a reward or not, even in cortex. And that's something that we think has been overlooked. And so we're trying to move that to that realm. But
Okay, awesome. Last point, if you go back to your model, that SST, you had SSTP and N, and SSTP was the one that was responding. How did
to
you
reward, yes.
To reward.
We don't know why. I have no idea why. We don't know why these neurons are responding to reward at all. What I can tell you is that they don't respond to acetylcholine inputs. The dynamic doesn't match the dynamic of acetylcholine release. We don't really know what's going on there. Okay, so Rui Costa had this like hierarchical model that actually comes from the PFC and then is being backpropagated throughout cortex, and this backpropagation could happen through somatostatins. But yeah, so.
Okay, very interesting. And Rui Costa, you mean Alan Rui Costa or the computational Rui Costa?
Computational in Oxford,
yeah,
not Princeton.
And I like I always remember if you go to the one that shows the Campalin, or like the connectivity, if you go to that slide.
Like that?
Between VIP SST, no, the one that you had it for both, because I always remember them. Yeah, so I always remember the Campanile, I don't know how to pronounce his last name, work, but so that is also pretty cool. So those guys also did some circuit level like stimulation and recording, single cell.
In this paper?
In the Pfeffer paper.
I mean, in this paper, it's there's a lot of single cell RNA, and then probing the connectivity by like just paired recordings.
Yeah.
I don't know, but again, I think the I mean, and correct me if I'm wrong, but the Allen is, especially in this data set, is very interested in like local coding and running sometimes.
Campanula work was more, so that was the one that they do paired recording stimulation. That one didn't have like the other one is the visual behavior, so those are separate data sets.
But I think, I mean, there's the other part is like in this data set, actually, at least in the visual cortex, this is in visual cortex, but it's going to change. We're actually going to move to parietal cortex data. The depth location of this like responding, not responding, I'm not sure. I would not make I would not bet a lot of money on whether this is this direction or the other direction. I think Campagnola says it's the other direction. So yeah.
Meaning that you are also using P and N, but by the N is that I, right?
Yes, sorry.
Right. Yes. So you guys found more superficial SSTs not responsive, and you're saying Campagnola says
that if this responding not responding corresponds to the bidirectional VIP somatostatin, would find the other way around, and that's kind of what we find in the PPC actually.
That what? I missed this.
That whether you respond or not to reward is kind of a functional indication of whether you're connected or not to VIP neurons and the direction of the connection. And Campagnola would find the other way around. But again, it's like this is layer 2-3, and we are actually mostly recording in layer 2-3. So we're trying to like separate neurons within almost layer 2-3. So I think, there's our, this is not this is not gospel here. Like I would, this could change. Awesome,
very insightful. Thanks, Quentin. We're 15 minutes over time. Do people have any questions? Guess not. Thank you, Quentin. Much appreciated.
Thank you for listening.
Have a good day. Bye.
Bye. You too.